Let's go. Yeah. Oh, I mean, it work. Last year.
Okay, sorry about that, folks. Couple emergencies down the hall. All right, so when we left off, we were talking about the membrane potential of neurons. So we talked about resting membrane potential, talked about the fact that for neurons it's at -70 millivolts. We talked about how that gets set up, what that means for sodium and potassium specifically, and how the behavior of sodium and potassium really gets us to that point as potassium pulls us towards its equilibrium potential of -94, trying to escape the cell, pulling out positive charges. Sodium is trying to get us to +60, but the problem is we don't have very many open channels for sodium to go into the cell, so it's not replacing all the potassium that's leaving. Plus, we know that we have a sodium-potassium pump. It behaves in an electrolytic manner, meaning we're pulling an extra sodium out for every 2 potassium that come in. That's setting up a concentration gradient that leads to those equilibrium potentials for sodium and potassium, but also it just physically removes the sodium every time it does it, pulling that membrane potential down. So we walked through what's going on with sodium, we walked through what's going on with potassium, and we're left off kind of right. So for our resting membrane potential, we're at -70. Remember that that's pretty close to our equilibrium potential for potassium of -94 millivolts. We remember that an equilibrium potential is what membrane potential you would have when all of our forces on that specific ion equal out, cancel each other out. So at -94, that were the membrane potential of our cell. There'd be no net movement of potassium because our chemical driving force and electrical driving force on potassium would cancel each other out. Okay. So we can see that at the resting membrane potential of -70, we have an electrical force into the cell, right? So we have those positive charges attracted towards that negative charge in the cell. But we have a chemical force out of the cell because we've used the sodium-potassium pump to set up these strong chemical gradients where we have lots of potassium inside the cell, not very much outside the cell. So because we're quite close to the equilibrium potential relatively, this is a relatively weak total electrochemical driving force on potassium. So potassium does want to flow out of the cell. Our electrochemical driving force points out, but this is pretty slow. We have slow flow out of the cell, but we have lots of open channels for potassium, unlike we do for sodium. So that high permeability keeps us pretty close to our equilibrium potential for potassium anyway. We got the opposite situation for sodium. Sodium's really far from its equilibrium potential. At our resting membrane potential, both our electrical and chemical forces come in. Yeah.
Is that a plus or a minus, or is that an underlying plus?
Oh, okay. Sodium is a positive ion, so I think it's an underlying plus. Sorry about that. That's interesting. It underlines the superscripts as well. Thanks for that. Yeah, sodium's always positive. Okay. Yeah, so we're far from sodium's equilibrium potential. When we're negative, both chemical and electrical driving forces point in, which means we have a really strong force for sodium to move into the cell, but we have low permeability to sodium, don't have any channels for it, so we don't actually have that much movement of sodium into the cell. And we can see that difference is actually 130 millivolts apart. So our neuron at rest does have leak channels throughout. So the channels that we've just been describing as high permeability to potassium, low permeability to sodium are going to be described later on as our neural channels, maintaining this resting membrane potential when our neuron is at rest. So we have a small sodium leak into the cell. We have a relatively small potassium leak once we're at -70, right? A bunch leaked out to get us to -70 if we started at 0, but once we're at -70, not too much potassium is trying to escape. There's a relatively weak force on it, even though we have lots and lots of open channels for it. And we've got this sodium-potassium pump, keeping those sodium and potassium gradients relatively stable because those equilibrium potentials have those specific gradients kind of built into them. So you can see a short clip of this happening in L2D on a video, I guess, but see what's going on.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at -90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to -70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
So that's how this overall gets set up. Good to understand, but what we want to focus on is making sure that we understand the things that are true about our neuron at rest. The fact that it's at -70 millivolts, the fact that that's pretty close to the potassium equilibrium potential, which is at -94, really far from our sodium equilibrium potential, which is at +60, leading to a strong force for sodium to want to come into the cell, and a weak force for potassium to want to flow out of the cell. Okay. And the reason we're setting this up is that as we eventually get towards describing our action potentials, so describing how we actually get a signal coming from the body of our neuron and flowing through the neuron to some other part of your body to send a signal, as our action potential, we're going to see changes to the membrane. So we're going to see changes to that electrical charge on the cell. So we're going to see specifically something that looks kind of like this. So we are going to see that starting from rest, in order to fire off a signal, so here's our action potential, the spike of charge is really what's going to tell the neuron at the other end to release like neurotransmitters to whatever cell it's talking to. All right, we're going to have an initial depolarization. So we're going to get a little bit closer to zero. We're going to get to something called a threshold. So that's going to be at -55. These numbers will be super familiar to you by the end of the unit, but we're going to get to here to a threshold. This is going to kick off new behavior in the cell. It opens up a ton of sodium channels. We've already set up this neuron such that we already know that sodium wants to flow into the cell. When we get to this threshold, we let it, which is why we see this spike up to +30 before we see another change in the membrane of a neuron that pulls our membrane potential back down, kind of overshoots threshold a little bit before we stabilize back to rest. So we'll talk about all of that in detail and what's happening in these different phases, but that's where we're going and why we're trying to understand what our initial kind of baseline state is for our neuron. As we are talking about neurons, we'll be discussing specific locations. I'll just run you through this, but you should be able to name the parts of a neuron on your own after class, so do practice with this later on. So as we're talking in this next part, we're going to be describing, okay, like what's going on in the body of the cell, what's going on in the axon, et cetera. So I want to practice with that. Okay. So when we're talking about the structure of a neuron, we're going to refer to like the main lumpy part where you see the nucleus and the organelles, this part, as the soma, which is just, I think, Greek for body. Maybe it's Latin, but I think it's Greek. So sometimes I will say body interchangeably with soma. This is the part we're talking about when we're talking about the soma. When we talk about dendrites, we're going to be talking about these parts of the neuron. They're linked up to the soma, linked up to the body, and are going to be receiving incoming signals. So where you see these green arrows coming in, this would be a dendrite. So this is like the receiving part of our neuron. So you can see the charges that might accumulate in the dendrites would flow here and come into the soma. On the other side, we're going to eventually be thinking about talking to another cell over here. Right? We're going to have another cell, cell 2, okay, which might be another neuron, might be something like a muscle cell, a heart cell, a gland, whatever it happens to be. Okay, so cell 2 over there eventually. So we're also going to be thinking about these signals flowing out. So you're going to see this pinching off point here from the body of the neuron, from the soma. This little triangle is what we're trying to focus on here. You think of it as looking kind of like a hill, and a triangle shape to it. So this is the axon hillock. Okay. So eventually we're going to be focusing on this location as the starting point of our action potentials, because our action potentials are going to be going down this long, skinny axon. So the axon is this part, sending us out. Okay. Our action potential will be here. Okay. And then eventually we'll have the release of signaling molecules called neurotransmitters. Those are going to come from the axon terminals. So terminal, think terminal illness, think end. Terminal is the end point. Here. Sorry, guys. Hopefully that's not triggering. Okay. Terminals. Axon terminal is the end. Okay. So we've been drawing little circular cells to this point, so we can make an analogy kind of to the soma, to the body of the neuron, but as we talk about other types of channels and what's going on as we propagate, send our action potential down the axon, we're going to be focusing in on other parts of the neuron as well. Oops. That's supposed to be next. Just want to make sure that I'm going. Oh, okay, cool. There's another secret topic nested in before we get to the action potential itself. So I wanted to make sure I wasn't skipping any packets for you. Okay, so we're in the action potential packet. Okay. So our overall goal for this next chunk is to think about how we get from the soma, from the body of the neuron, to actually sending a signal all the way down to the next cell in line, sending neurotransmitters out of our axon. So to do that, we're first going to talk about a slightly different type of potential. So we've talked about membrane potential so far, so that existing state of the cell at rest, -70. We talked about equilibrium potentials, this kind of theoretical membrane potential where the movement of a specific ion is net zero. Now we're going to add on graded potentials. When we think about graded potentials, you want to think about this as kind of like, this is going to be a yes/no question for us almost. The graded potential is something that's going to build up in bits and pieces. Our ultimate goal is going to be to figure out whether our graded potential is big enough to make the cell fire off an action potential, so this big signal, or not. Once we've talked about both of those, we'll have talked about all our different types of potentials that we have in this unit. The graded potentials that I wasn't sure. So we gotta talk about those. So what a graded potential is fundamentally is going to be like a small change to our membrane potential in kind of a local area. So when we're thinking about our neuron, I draw, this is like my soma and body. These spiky bits are going to be my dendrites. And the soma, then we have our axon hillock, then we're going to have our axon, and eventually some axon terminals. The lazy neuron, but different than a circle. Right? We're going to be thinking about graded potentials as collecting here through these incoming signals to the soma, to the body of the neuron itself. So coming down dendrites or occasionally striking the soma, the body of the neuron itself, leading to some short communication, some small changes in this kind of local area that eventually are either going to depolarize this axon hillock, so actually get us from -70 to our threshold of -55. Okay, so they're either going to make that change at that spot or not. And if we see that change to -55, then we're going to shoot off an action potential down the axon. If we don't get up to -55, the graded potential is going to fade away. So that's kind of like the big picture purpose of talking about graded potentials, is these have to happen first. They're kind of small. They've got a little more variability to them than the action potential, which is always going to look like that specific graph that we saw with the big spike to positive 30. Okay. So here we see an example of a possible series of graded potentials. All right, graded as compared to an action potential. That's what we have here in B. So you can see what we're tracking in both of these graphs are changes to our membrane potential, right? So changes over time to that charge in the cell. And we can see that our graded potential has some small little lumps, got some small dips, nothing really big. Our action potential is always going to have a rise to negative 55, a spike to positive 30, and then a huge dip before coming back to rest. So always going to look like that. Graded potentials are not always going to have like two lumps and then one dip and be any kind of color. So comparing these two, we can notice that our graded potentials are smaller. And when I say small, you're thinking about what the change to the membrane potential is. That relatively small integrated potential or action potential has that big spike. Okay? So that's the large part of the action potential. And when we talk about distances, we're talking about the distance along the neuron. So when we're thinking about graded potentials, we're going to be thinking about not really the whole cell's membrane potential changing, but just little parts of the cell. When we think about the action potential changing, we're going to be thinking about that change happening all the way down to the end of our neuron, which happens to have this long skinny piece called the axon. So we're going to have some communication over short distances and a graded potential, long distances for our action potentials. We'll do the questions that aren't shown up as a review next time, so stay tuned for those. Okay. We can just talk about sodium, so I think we're good. So remembering our setup, right? We had our resting membrane potential, right? Negative 70. We know sodium wants to come into the cell. We know potassium wants to leave the cell. That's our setup initially. So that's going to be our framework as we're thinking about graded potentials, how they collect, what they're doing. So graded potential is going to be a response to a stimulus, right? So to see a change in the membrane potential, the graded potential will be a change, a little change, a local change to the membrane potential. We have to see something happen. So that's our stimulus. So here we see our stimulus coming in to the body of our neuron, the soma here, some kind of signal coming in. So this might be a neurotransmitter coming in and touching the cell, coming from a different neuron. This might be some kind of physical touch or stretch that could also potentially act as a stimulus in specific locations. So there's something coming in that's affecting our membrane here. So some kind of change. Okay. That changes our channels and permeability of the membrane in that spot to different ions like sodium and potassium. Right, so the stimulus is coming in a little signal. In that little spot, we're going to see the membrane physically change in some way. And this is going to allow some sodium ions to come in or allow extra potassium ions to escape. Or other ions might also have an effect. So some type of ion, we're still thinking about charge, we're thinking about changes to membrane potential, which essentially is a charge difference across the membrane. So our stimulus is coming in, it's causing this change to the membrane potential at the location where it's poking the cell. Might be a big change, might be a small change. That's going to depend on the stimulus, and it's going to depend on what it does to the membrane, like how many channels it opens, how many channels it potentially could close. Okay. So you're going to see a big, potentially, change to the membrane potential right here where the stimulus comes in. But as we track the overall change towards like what's going on with the membrane potential here, or here, or here, you're going to see that there's a little bit of decay in that strength. So as you spread away from that stimulus, that location where the stimulus came in, like maybe we see a change of like 5 millivolts to the membrane potential here on this patch of membrane. But as we move farther away, maybe there's only a change of 3 millivolts by the time we get to here, only a change of 1 millivolt as we get over here to the other side of the cell. So we're going to see this decay. Right? When we say our graded potentials are decremental, this is what we're talking about. The fact that the strength of that graded potential, the strength of that change in charge, getting smaller as we move farther away from the location of the stimulus. So it might be a big or small stimulus. It's going to spread across the cell, but it's going to get smaller and smaller in its effect as we get farther and farther away. This spread is happening through electrotonic conduction. That's going to be our formal term. But essentially, when we think about changes in membrane potential, really what we're thinking about, how many positive ions do we have? How many negative ions do we have inside versus outside the cell? So you can think about those ions as kind of spreading out. More of them are kind of clustered together at the spot where we first let them in. And they start to kind of diffuse. A few of them end up over here. A few of them end up over here. So that's how they're spreading, if they're kind of floating away. And that's also part of why this strength is decreasing as we get farther away. Just by probability, fewer of them are going to get over to that. So the result is that when we record our membrane potential, at that point where we had our stimulus come in, it'll be a little bigger. Right? If we recorded our membrane potential here, right, we can see that the change to the membrane potential would be a little smaller. Right? So that's that decay piece. And we'd be paying attention to what stimulus it is. Right? So here's an example of a smaller stimulus. Here's an example of a bigger stimulus. Right? So we're really just talking smaller change, larger change there. So the point of thinking about graded potential, as we're thinking about the nervous system and neurons, is to figure out, is this neuron going to fire off an action potential or not? Because any neuron, think about your brain, going to be connected to like a lot of different other neurons, right? So they're all kind of firing together at the same time. They might have conflicting signals. They might be saying the same thing. And any individual neuron is going to have to figure out in response, okay, am I going to fire off my signal now or am I not? What do I do with all this incoming information? So that's what we're thinking about when we think about graded potentials. We're thinking about basically adding together inputs to make this binary yes-no decision about firing off an action potential. So that's where our threshold comes in. I say deciding, that's how I think of it, but really what's happening, the decision is kind of a mathematical decision. We're thinking about, do we reach this threshold value, negative 55 millivolts, or do we not, right? If we reach our threshold of negative 55, we'll fire off an action potential. If we don't, like if we get to negative 60 millivolts, graded potential, but we're not going to fire off an action potential. Okay, so we might have stimuli, we might have incoming signals, but they wouldn't be enough to get this neuron specifically to do something in response. Our threshold is negative 55 millivolts. We'll be on lots and lots of later slides, but I'm going to repeat it as many times as possible so that it's easy to remember later on. Okay, the threshold is negative 55 millivolts. Counts as a depolarization because we're moving closer to zero. So we're going to think like in terms of the number line. This graph we've been seeing, right? We've had negative 70 here. Let's imagine 0 here. Imagine positive 30 up here. Okay. So our formal word for describing the sign on that membrane potential, right, whether negative or positive, was the cell's polarity. Right? So polarity plus or minus on the cell, on its membrane potential. So when we depolarize this cell, what we're saying is we're removing that polarity, which is really just describing that direction towards zero. So depolarization is this direction. That's depolarization, describing a direction of change to the membrane potential. So some of our graded potentials will be depolarizing. We're checking whether they are depolarizing enough to hit -55, which is our threshold. Okay, so that's our question. Did we depolarize or go up enough to hit -55? If we do, we fire off a big action potential, which will have a bunch of named phases that we'll talk about later on. We'll refer to depolarizations like this as excitatory. Reason for this is the cell is getting excited if we're depolarizing, right? Like we're getting ready to do something. If we hit the threshold, if we depolarize the threshold, we get to fire off our action potential. We get to send this electric signal down the axon. We get to send out our neurotransmitter. That's exciting. So excitatory graded potential would depolarize our cell a little bit. That's not the only thing that can happen. So to depolarize, we need to let positive charges flow into the cell. We're starting at -70, make that more positive. That fundamentally means positive ions coming in. But we could have negative ions flow in. We could have positive ions flow out. That's possible. We already know that potassium wants to get us down to -94 millivolts. We're not there yet, so we could get more negative. That's a possibility. So we have another word for that. So if we go the opposite direction from -70 and make this even more extreme, that's our hyperpolarization. Right, so hyper for extra. So hyperpolarizing down. And if we get down below -70, it's going to be even harder to fire off an action potential, right? If you start to have more potassium flowing out of your cell in response to a stimulus, if you get extra negative, then if you want to fire off an action potential, if you want to send out your neurotransmitters, you're going to have some other stimulus come in that's really big to make up for it. So that's why we refer to our hyperpolarization as inhibitory. So being inhibited, like not wanting to do stuff, that's this direction, the hyperpolar direction. So you can see them on this graph here. Right? So here we have stimulus Y, which is hyperpolarizing. So here's our graded potential in that hyperpolarization direction. Here we see a depolarizing stimulus leading to this excitatory graded potential. So going up, getting us closer to threshold. Perfect. Okay. So graded potential are these small changes in response to a stimulus in a specific part of the neuron that spread out to get weaker as we get farther from that point where the stimulus came in. We're describing them as a decision to fire off an action potential or not. So part of that decision is we're going to be adding stuff together, right? This is a way to handle multiple inputs, multiple stimuli, and kind of look at that in aggregate, sum it together, figure out what to do in response to a bunch of different incoming information. So there are two ways that we can add up our graded potential. So summation could be a word for adding them together and figuring out, okay, so we have one stimulus coming in, it's depolarizing, so it's getting us closer to threshold. But we have another one that's hyperpolarizing coming in at the same time or just right after. Where do we end up? Right, so summing them together, that's what we're talking about. When I say different ways, what I mean is that we might be trying to handle two signals coming in rapid suggestion. So think about someone like poking you once and then poking you again a bunch of times really fast in a row. That's going to be our temporal summation. We handle those things differently. We're doing slow pokes versus fast pokes. The other thing we might be summing is spatial, right? So imagine you have 4 people standing around you and they all poke you at once. That's different than if you just have one person poking you. So we also have spatial summation. So we're not talking about like math being different, addition being different. We're just talking about, are we adding together signals that are close together in time or temporal? Or are we adding together signals that are close together in space? And that's our spatial summation. So on these examples of memory potentials, we can see individual graded potentials happening over time, right? So this is 2 different stimuli, each of which causes a peak. We can see for this third arrow, we've got a larger change here. So it might be the case that we had these 2 stimuli from earlier on happen in super quick succession, getting us to threshold when we add them together. So that might be more like temporal summation. We might have spatial summation where they happen at exactly the same time. So let's think W plus Y here. Say they're coming in on opposite sides of the cell at the same time. We add them together. Might end up closer to threshold, or sometimes we'll see that our graded potentials that we're adding together that we're summing are in opposite directions, so they might cancel each other out too. These are both possible results. I'm going to draw a couple of examples for us of how this works. Okay, so we're going to be thinking about neurons at 3 kind of points in time. Okay, so I'm going to draw some dendrites, an axon. There you go. More cartoon character looking as we go on. Okay, so we're going to have 3 neurons. It's really the same neuron at 3 points in time if we're thinking about temporal summation. Okay. So we'd be thinking at time 1, we have a signal come in. So this is our stimulus, right? And say this causes a slight depolarization to our membrane potential here. So let's say that instead of getting to -70, or sorry, instead of getting to -55 from -70, we're going to get to -60. Okay. Okay. So this is our stimulus 1. Then say we have a little bit of a slight rest period. So a graded potential is going to dissipate a little bit over time. So after that stimulus, we don't fire out an active potential, we don't do anything. Maybe that's going to go from -60 to like -65. It starts going back down. Okay. If we then have another stimulus come in, that same stimulus, okay, so this same stimulus caused a change of 10 millivolts from rest, right? So we went from -70 to -60. Now let's say we get this signal again, And we haven't gotten all the way back to our resting membrane potential. So stimulus 1 is coming back in. It's going to have the same effect as before. So we're going to change it by 10 millivolts again in that depolarizing direction. All right. So now we're going to be at -55. Right. So that's one way we can be adding together conceptually our stimuli over time as this happens twice in a row. So this is one way we might get to threshold. Okay, so now we're at threshold and we fire off an action potential. For spatial summation, we're gonna have something a little different. But our question is the same. Our question is, do we get to -55 or not? That's still what we're interested in. I can draw a bigger one because we're not worried about time. So we don't need to have 3 anymore. Just gonna have 1 neuron. We're going to add together in space. So we had before our stimulus 1 coming in and causing a change that gets us to -60 over here. Right? It might be the case that we have another change, another stimulus happening over here, right? So spatially, we might have 2 signals coming in at once. Okay. So if we had only 1 stimulus coming in at once, we'd have some decay over space as well as decay over time. So our action potential is going to fire off down the axon, so the skinny part. So what we're interested in is, does this specific patch of membrane hit -55 or not? That's our axon hillock. It's the beginning of our axon. So this is really the location where we care about the charge. So because gradient potentials decay over space, right? So let's say we have that -60 over here. Let's say that as that charge spreads, it gets a little weaker. So maybe it got this patch of membrane to -65 in this location, right? So not quite that 10-point change, just a little 5-point change since we're farther from where this is. But we have another signal to contend with, right? So we have signal 2 as well. So let's say signal 2, that same strength. Right? So let's say it also gets this to -60. So we also have a 10-point change right here at that location. But if we're focused on the effect over here on the other side of the cell at the axon hillock, we're going to have to think about all the ions that have come in from this second location, right? So this is essentially a bunch of ions coming in and then spreading out, right? So was what's happening here at our first stimulus, right? So we got a couple of those ions floating to the side of the cell from stimulus 1. We also got a couple coming from this stimulus 2, right? So this is the second type of summation. So if we're trying to figure out what's happening at the axon hillock specifically in this situation, we're not going to be just at -65, right? Because this stimulus 2 is also depolarizing us a bit, right? So we might add together these 5-point changes. 5 from S1, then 5 from S2, getting this to -60 in that axon hillock. So this one wouldn't get us all the way to threshold using these fake numbers that I put in here, but you can imagine that if one or both of these stimuli were a little stronger, we could similarly get up to -55. Questions about either of these types of summation? I know sometimes people get a little lost in the weeds on this. Usually it's the case that it's actually simpler than you're thinking, but I want to give you space to ask any initial questions about that. So, big thing is that we're adding up these small changes, trying to get to thresholds or not, might be adding them together close together in time, might be adding them together because they're coming in different parts of space. This is a good place to stop, and next time we'll be thinking about how these ions are actually working to cause an action potential, and we're going to think about voltage-gated channels. I do recommend taking a look at the figures for voltage-gated sodium and potassium channels, but really the sodium ones before class next time. That's another topic that can be tricky the first time you see it.